Thank you for joining us today. Hello, Merritt. It's always nice to see you. And so our guest speakers today is Dr. Merritt Cudkowicz and also Dr. Sabrina Paganoni, who are the principal investigators at the HEALEY ALS Platform Trial. And we have exciting, promising drug, Amylyx ALS drug that all of you have heard it many times. And we are excited to have some of the promising results discussed today. And I think we also have Joshua Cohen and Justin Klee, founders of Amylyx, who are joining us today as well. And thanks to the young mind and brain who have actually been part of the ALS community, we are grateful to the Amylyx startup here. And with that, I would ask Merritt and Sabrina to take it from here. Thank you.
Excellent. I was really, when you said young minds and brain, I thought you were talking about Sabrina and me, but.
That too, that goes without saying. It wouldn't happen without you guys' mind and brain. So thanks for that.
Sabrina, do you want to launch?
Sure. Thank you, Merritt. And wow, that's a fantastic community. I really appreciate the opportunity to talk to all of you tonight, and especially to share this presentation with Merritt, as well as Justin and Josh, who will join us for the panel. I'm excited to share the results of the Center trial. Many of you have heard about it before, but tonight we really want to have an in-depth conversation. Feel free to ask any and all questions. We're glad to share the exciting results of this trial and the promise of AMX0035 in terms of function, safety, and survival for people with ALS. If we can go to the next slide, I would actually ask Mary to start and tell us about the story of this trial.
Yeah, thank you. This is such a great story, and it really shows how a couple things. One is, like Indu said, the importance of young minds and new ideas entering the field, but also how welcoming and open our field is to the ideas, no matter where they come from, and set up the resources and the teams to help anyone who has a good idea for ALS and a passion for ALS. So the idea for the AMX0035 came from Josh and Justin while they were undergrads at Brown. They can tell you more about this than I can, but they were really thinking about cell death. Now, I'll say I don't think my daughter, who was going to Brown, was thinking about cell death when she was in her dorm room and her friends, but you guys were, which is fantastic. And thinking about also repurposing drugs and how you can make a difference, and really came up with this idea that combining these two agents that work on different parts of the cell, one on the mitochondria, which are the parts that make energy, and one on the endoplasmic reticulum, which is part of the cell that helps with misfolded proteins or protein clearance, that maybe together they would be better than separate. And they launched a company from like they said, from their dorm room. And they reached out to a lot of people, but one of them was Dr. Rudy Tanzi, who is an amazing Alzheimer's scientist that works at Mass General Hospital. He's also a rock star and does a lot of other things, but really a genius. And he met with them and they talked science, they gave them some advice. And really, to be honest, I met them by chance. I was going with Peter Foss, who's the president of ALS Finding a Cure. We were going to meet with Rudy Tanzi in Charlestown, where his lab was, to talk about the foundation. He was a chief scientific officer for Cure Alzheimer's, and the foundation I was a chief medical officer for, ALS Finding a Cure, whether we could co-fund some work together in Alzheimer's and ALS, where there were some overlaps. And Josh and Justin happened to be there at that time. And so he said, come meet these two. And so we started to talk, and really literally, I think at that meeting, the decision was for those two organizations actually to co-fund some critical preclinical tox studies that needed to be done to bring this to patients. Now, the other reason I was excited about it is I had actually done a study of the phenylbutyrate in 2008 at the VA. I actually got a position at the VA just to do the study because they were going to fund it. And we did a phase 2 study and we found a good dose. But the FDA at that point wouldn't let us go any further because even though that it was a marketed drug, there had been no toxicology or safety studies done in animals. And to go into bigger studies, they required that. At that point, the drug was generic and there wasn't anybody willing to do it. So fast forward, Josh and Justin had found a good rationale, not just for phenylbutyrate, but for the combination. So then they did those studies and they applied also to something, a program called ALS ACT, which was a program we started between ALS Finding a Cure and the ALS Association to fund early phase trials. And actually that program has been really quite successful. It funded the new DEXTA study Richard Smith did to show that drug helps swallowing and speech. Funded the Amylyx one, and it funded the first gene therapy in SOD1 and so on. So that's a real big program, and I hope those two foundations will continue it. And then Sabrina came in and the NEAL Scientific Advisory Board to really help work with Josh and Justin and Amylyx on how to design the trial and to conduct the trial. And fast forward now, we're at a positive trial, which is really quite exciting. So that's the history. Again, I'm just thankful that we've gotten to be part of it and gotten to learn a lot from Josh and Justin and their companies and vice versa. So with that, I'll stop talking and let Sabrina really dive into the actual results with you.
Great, thank you, Merit. And we can go to the next slide. And again, I just want to emphasize once again that the Center trial was a true collaboration in the ALS community, and how wonderful it is. It was for me when I started. It was my first trial actually as a new faculty at NGH. I was really fortunate to be mentored and to learn about clinical trial design from Dr. Cudkowicz. And then this amazing opportunity came to partner with Amylyx, as well as 25 phenomenal centers of the NEALS consortium to be able to run the trial and enroll participants from many different centers that are listed on the slide. And we coordinated the trial from Mass General and the Barrow Neurological Institute, because we have the project managers, data managers, and an entire infrastructure to not only design trials, but also to collect high quality data and really collect data that can be used again for really demonstrating the efficacy of new drugs like AMX0035. And it's been wonderful to receive some of the initial funding from the ALS Association, ALS Finding a Cure, and we got a lot of input from the patient community in terms of the design of the trial and ways to advertise the trial and connect to the patient community. So it's really been a phenomenal journey. We designed the trial in 2015, 2016, and we actually included a lot of innovative aspects, many different features that nowadays are sort of, taken for granted. But really, when we designed the trial in 2015, 2016, things like an open label extension or other features of the trial, doing webinars, outreach to the patient community, including patients as part of the steering committee. All of those things, we included, and I'm so glad that, again, it was such a collaborative, project from the beginning. Next slide. And now we have a short video. I know there was some. Okay. I hope that you can hear. I saw some conversations about the volume, so just wanted to make sure Indu and the organizers, Lisa, are we all good with that? Okay, so let's go.
In ALS, multiple genetic and non-genetic insults and risk factors cause motor neurons to malfunction, degenerate, and die. Although the relative contribution of each factor may vary among individuals, many of the pathways that lead to degeneration in ALS converge on the mitochondria and endoplasmic reticulum, or ER. Mitochondria are the powerhouses of the cell. They are responsible for maintaining metabolic requirements, ensuring calcium homeostasis, and may trigger apoptosis, or cell death if necessary. In ALS, mitochondrial dysfunction, release of cytochrome C, as well as an imbalance of pro- and anti-apoptotic factors trigger the mitochondrial apoptotic pathway, leading to cell death. The ER is the key site of synthesis and folding of virtually all proteins that transit through the secretory pathway. In ALS, ER dysfunction in combination with other abnormalities leads to aberrant protein folding and the accumulation of toxic protein aggregates. These aggregates cause stress on mitochondria and overload the unfolded protein response, resulting in a feedback loop that leads to more ER stress. In addition, normal crosstalk and homeostasis between ER and mitochondria are disrupted, further driving dysfunction. Together, impairment of mitochondria and ER function contributes to degeneration and death of the neuron. With the continued loss of motor neurons, muscle function and vitality progressively decline, diminishing independence and quality of life.
Yeah, I think this was a beautiful illustration of the mechanism of action of the drug, of AMX0035, which again is a co-formulation of PB and terso. Now, the interesting thing is that PB and terso, as the video showed, target different cell components to keep the nerve cells functioning properly. Individually, as individual compounds, they were previously shown to have neuroprotective properties in different lab models, including lab models of ALS. And so the interesting change here is that Amylyx Pharmaceuticals decided to combine the two in the lab, and they found that the combination had even better effects in the lab. So based on those results, we were excited to work with Amylyx Pharmaceuticals to test the effect of the combination in people with ALS. And the results that we're showing today were obtained by specifically using AMX0035, which is a quality controlled and optimally formulated combination of the two individual drugs. Next slide. Next, yeah, thank you. So AMX0035, as used in the trial, is this proprietary co-formulation taken by mouth or via feeding tube. It's packaged as a powder in a pouch and can be dissolved in water. So it was taken by the participants once a day for the first 3 weeks, and then twice a day. It can have a bitter taste, and so we did match placebo to the active drug for both appearance and taste. Next slide. So what does CENTER mean? Well, it's actually an acronym that's derived from the sentence that you can see on the slide. The sort of the short title of the trial was, the trial to test the combination of phenylbutyrate (PB) and taurursodiol or terso. So that's how we got the name for the trial, CENTER. And we started enrolling in the trial in 2017. And then we finished, patient follow-up in late 2019, and we published the main study results in 2020. Specifically, in September, we had an article published in the New England Journal of Medicine to report the effect of the drug on physical function. And then in October, we had a second paper published in the journal Muscle and Nerve that reported the long-term survival results of the trial. So I do want to point out that we also gave previous webinars on these two separate papers. And so while today we're combining all the results, if you want more information, you can also always go to the NILS website that's on the slide to watch the actual recordings. And there was also always a robust Q&A section after each webinar. So you might find those interesting as well. Next slide. So the CENTER trial was made possible thanks to the generous efforts of 137 participants with ALS. When they enrolled in the trial, their average age was 57. On average, they were at 13.5 months since symptom onset, and they were about 6 months since diagnosis when they enrolled. About a quarter of participants had bulbar onset, and as you know, bulbar onset means onset in the muscles that support speech and swallowing, so they might have had speech and swallowing difficulties as their first symptom. And the vast majority of participants were taking riluzole, edaravone, or both at study entry Specifically, 77% of participants were on at least one of these two medications. And as you know, these two medications are considered a standard of care, so people can, in the clinic can freely choose to take them. And we really didn't want to restrict access to standard of care. So we really allow participants to take these drugs if they wanted to do so. So the results of our trial of the study of AMX0035 are not a head-to-head comparison to other drugs, but the results that we are seeing and the positive effects are on top of standard of care, because the vast majority of participants were already taking those medications. Next slide. So thanks to these 137 participants, we were able to test and answer several questions. So we wanted to test the effect of AMX0035 on physical function, and I'm reporting the results over a 6-month period. I will explain the design in a second. We also were able to test the effect of AMX0035 on survival. This was assessed long-term, up to 3 years after the beginning of study participation. And of course, throughout all of these, we also wanted to confirm that AMX0035 is safe and tolerable. Of note, the individual components had been tested in previous trials, and individually they were safe. But again, we wanted to make sure that this was true for the combination as well. Next slide. So I told you that the first question we wanted to answer was the effect of AMX0035 on physical function. And now, for everyone to be able to understand the results of the trial, I just want to review how we measure physical function in ALS. I'm sure most of you are familiar with the ALS FRSR. This is a questionnaire that's oftentimes administered in clinic or as part of research. And it's a series of questions, 12 questions, that measure independence with several activities. And so, for example, in order to test someone's bulbar function and independence with bulbar function, we ask questions about speech, salivation, or swallowing. And then to assess fine motor skills, we may ask questions about handwriting, for example. Gross motor skills are assessed by asking questions about walking, for example, and so forth. So essentially, we ask a series of 12 questions asking about activities that people do in their daily life. And we ask them whether they are independent or not on those activities. Now, a score is then assigned to each of the 12 questions. And the scores range from 0, which means function absent, to 4, which means function normal. And so essentially, when we look at the total score, the total ALS FRSR score gives us basically a snapshot of someone's independence. And the score goes from 0 to 48, where 48 is normal. Next slide. So I want to explain the trial design. So center included both a randomized placebo-controlled period and an open label extension. So at the beginning of the study, we randomly assigned 137 participants, 2-to-1 active to placebo. This means that the active drug group was twice as big as the placebo group. And so essentially we had 89 people on AMX0035 from the beginning, and then 48 people on placebo initially. Again, and their participation in this randomized period lasted 24 weeks, or about 6 months. And over this period, we carefully measured safety and function in all the participants. Then we also had an open label extension. So what this means is that at the end of the placebo-controlled period or randomized period, every participant who completed the study was given the option to receive the active drug long-term. And this is true regardless of their previous assignment. The open label extension was optional, and of note, of the people who completed the randomized period, the vast majority, 92%, opted in. So what this means is that people who received AMX0035 during the first 6 months of the randomized period could continue taking the drug long-term. And then the people who started out on placebo and took placebo for 6 months, then they were given the option to switch and receive active drug long-term. So then we followed everyone long-term to assess the effect of the drug on survival long-term up to 3 years. And I just want to make sure that we all understand that we checked survival on everyone, on all the 137 participants, so not just the ones who entered the open label extension. Next slide. And here I'm going to start by showing the first section of the results, the effect on function. So treatment with AMX0035 significantly slowed down disease progression as measured by the ALS-FRSR total score. So I told you before that the score basically assesses independence with activities of daily living. And you can see here AMX0035 in red at the top and placebo in blue at the bottom. As you can see, people who started out on AMX0035 were better at all time points. And there is a difference between the two, between the progression of the two. Now, both groups progressed, so the drug couldn't stop completely the disease. However, there was a significant difference in terms of their rate of progression. Next slide. And just to put this in terms of absolute numbers, what this means is that participants who received AMX0035 had slower progression than those who received placebo over a 6-month period, which corresponds to a difference of 2.32 points at the end of the 6-month study. So essentially, what we're saying is that by the end of the randomized period, people who were started on AMX0035, the people who received active drug, had a score that was 2.32 points higher than the people on placebo. So this means that they retain their functions longer, because as I told you initially, the total ALS-FRSR score, the higher the score, the better, meaning the more active or more independent with physical functions one person is. And so the people who received AMX0035 had their scores that were 2.32 points higher than people on placebo. Now, I do want to say that this particular calculation that I'm showing here is based on a very conservative way of assessing the differences between the two groups. And this was pre-specified when we originally designed the trial. So when you design a trial, whenever you pre-specify your analysis, then you sort of have to go with that as your primary result, because that's what you did before looking at the data. Now, the interesting thing is that both the FDA reviewers and the reviewers of the paper actually thought that our method was too conservative. So they asked us to do a different type of analysis that's shown on the next slide. They asked us to use a different statistical method that's called change from baseline. And when we applied the methods that they requested, the difference between the two groups was actually more pronounced, almost 3 points higher in the people with AMX0035. So what's the difference between 3 points, on the ALS-FRSR? Well, as you know, every point means more independence with functions. So that means that people were able to retain longer function, again, on overall on the scale. And the difference, the absolute difference was 3 points. Next slide. Now, we also did measure other things, secondary outcomes, such as we looked at muscle strength by using a device called Amylyx, and we also looked at respiratory function using a maneuver called slow vital capacity, or SVC. Some of you might be familiar with those measures in clinic because they are oftentimes captured in clinic as well. And as you can see here, when we compare the two groups, AMX0035 to placebo, all the results were better in the AMX0035 group. They did not reach statistical significance, but they all trended in the right direction, with AMX0035 being better. Now, I do want to remind everyone that the trial was powered to see a treatment effect on the primary outcome, which was the ALS-FRSR total score, the one that I just showed, that was statistically significant and clinically meaningful. We were not powered to see changes in muscle strength or respiratory function, because again, this was a relatively small and fast trial. So we didn't see a statistical significance there, but all the results go in that direction. I also want to point out, because I see some comments in the chat about sort of the difference in upper body or lower body. So as you can see here, when we express muscle strength, we express it as Amylyx scores, which is basically an instrument to measure muscle strength. And you can either combine upper and lower extremities in the total score that's at the top, or you can compute the upper extremity score and the lower extremity score separately. And those are the row number 2 and row number 3 in this illustration. As you can see here, the sort of the part of the body that was mostly affected in this particular calculation seemed to be the upper extremity score. But but that doesn't mean that that's the only one that was affected, because really we know from previous studies that that's a more sensitive part of the body to look at for longitudinal changes. So in a way, this was not completely unexpected. And I don't have it on the slide, but also when we broke it down, the ALS-FRSR total score into the 4 domains, checking the bulbar area, the respiratory function, the upper body, and the lower body, we saw something very similar, that the upper body function was mostly affected, was affected more than the other subdomains. And again, we know that that's the sort of the body area from previous studies that changes the most, and so it's the most sensitive to change. And I think that's why we saw a predominant effect there. What's important is that they all went in the right direction. So it seemed to affect all of the domains, even if, again, numerically, the upper body seemed to be moving more. Next slide. Something that's very important, obviously, whenever we are working on a new drug, is to make sure that the drug can be safe and well tolerated. So in the trial, the vast majority of participants had at least some adverse events during the study. Of note, this is very normal. We see that in every trial. We even capture normal disease progression as an adverse event. So again, this is not surprising. What's important is that there were no more adverse events in terms of general events in the AMX0035 group compared to placebo overall, if you just compute the number of events. Now, we do know that this small group of people in the AMX0035 group had some gastrointestinal events, meaning nausea, diarrhea, and stomach pain. And in the small group where this happened, it was mostly during the very beginning of the treatment, in the first 3 weeks. I also want to point out that we do capture serious adverse events, so the most dangerous events, separately. So these are sort of calculated separately from regular side effects. They are more serious, and so they are captured and reported separately. And those serious adverse events were actually more common in the placebo group compared to the AMX0035 group, possibly because of the more disease progression in the placebo group, and most of these were due to respiratory events. Next slide. So to summarize, so far I showed that AMX0035 significantly slowed down the progression of the disease. And because of these positive results, we wanted to see what happened to the same 137 participants if we were to follow them long term. So this is the newest data, the one that we just reported. And basically what we did was, to enroll all of the participants in 2017, 2018. Those are the people who participated in the randomized period. And after each of them finished their randomized portion of the trial, if they wanted, they were given the option to enter the open label extension, where they would receive AMX0035 long term. And the vast majority of people, 92%, actually opted in, decided to enter the open label extension. And then they could remain in the extension for as long as they wanted. And really, actually, some people are still on the extension as of today, and they continue to receive drug. So we started enrolling participants in 2017, 2018, and then we checked the results, the survival results in everyone on July 20, 2020. So this means that we followed them for essentially 3 years after their initial, entry into the study for the first participants who enrolled. Next slide. So to be able to tell whether AMX0035 had a positive impact on survival long-term, we compared 2 groups. So we compared those 89 participants who were originally randomized to AMX0035 when they entered the study to the 48 participants who were originally assigned to placebo. Next slide. So in order to measure survival in everyone, whether they're still on the trial or not, we used a variety of sources, including data from public records. And this is important because it was important for us to check survival on everyone, not just the people who are still in the trial. Again, we still have a few people in the trial, but some people might have stopped their participation. So we wanted to make sure to capture all their survival sort of in an unbiased way, using the same methods for everyone. And we did that by using a company called Omnitrace that is established in other fields of medicine, in cancer, etc., where they routinely use public records to assess overall survival from all-cause mortality. So we didn't capture if people were only died of ALS. We captured all-cause mortality from public records. And again, this is a very well-established and commonly used method in cancer research. And so once again, the cutoff date was July 2020, essentially 35 months or just a couple of weeks shy of 3 years after study entry. Next slide. So what we found was that the participants who were originally randomized to AMX0035 lived an average of 6.5 months longer than the participants who were originally randomized to placebo. So again, we're comparing essentially the people who started early to the people who started late. And keep in mind that even the participants who were on placebo, in order to have a patient-centric trial design, we didn't keep them on placebo the whole time. They were only on placebo for 6 months, and then they were given the option to receive active drug. So if anything, we are working against ourselves. In other words, we are underestimating the survival benefit because essentially we opened the door to everyone at some point, and they were able to have the option to take drug after 6 months into the study. And in fact, the vast majority decided to take it. And so that's why we think our results are probably an underestimate of the true survival benefit. I also want to point out that the effect of AMX0035 was independent of baseline use of riluzole and or edaravone. As I said at the beginning, we allow participants to take these drugs when they enter the study. So the results that I am describing today, both in terms of physical function and also survival, are not compared to standard medications. They are on top of standard medications, and that's very important because a question we often get asked is how does this compare to riluzole or edaravone, or will this be a substitute to those drugs? I think it's very important to emphasize that this is on top of those medications. So again, it's something else that we are adding to the treatment of ALS. Next slide. So yeah, and this is again that just to say that that was independent of those drugs. So again, to summarize, in the center trial, administration of AMX0035 resulted in both a functional benefit and a long-term survival benefits, and it makes sense. The two things should go hand in hand. So there was a significant slowing of the ALS FSR slope during the first 6 months in the randomized period, and there was also significant prolongation of median survival when people were followed long-term. And again, these results are seen on top of standard of care, meaning that participants could use the FDA-approved medications for ALS. And once again, the drug was safe and well tolerated over the long term, which is also an important thing to confirm, especially because people with ALS use many drugs for their symptoms and their disease over time. So it was important to see that we didn't see, again, any red flags in terms of safety and tolerability. And also, we didn't need to follow any lab. We did check labs and EKGs, but there was no signal. In other words, there's no need to check labs or EKGs if someone is taking AMX0035, because again, the safety profile is pretty good. Next slide. I do want to end by thanking the study authors. Many of you may know they're ALS clinicians and people who work at these excellent centers. It's really been a great collaborative effort. And many of them, again, have patients who are still in the trial, in the open label extension, and really great support from the entire community and the expert centers, especially MGH and the Barrow, in terms of study design and execution. contribution. And I also want to thank statisticians, both at MGH as well as Pentara and Statistics Collaborative. We had an army of statisticians who really looked at the data from any possible angle. And again, the results are incredibly robust and strong. And I also want to thank people at Amylyx. Really, it's been a phenomenal collaboration from day one. I particularly appreciate the fact that they really respect the academic process, the peer review process. It's really, again, a great collaboration. We're all trying to do the right thing and to really look at the data from every possible angle and just share the most robust and solid data. Next slide. And again, thank you to all the participants and their families. I don't know if there might be some on the line or people really this trial obviously would have never happened without their generous efforts. So thank you again, and I hope that we can have a great discussion with all of you. We're happy to take your questions.
Thank you, Sabrina. This was excellent. And the graphics and the presentation was just really, really easy to follow. So thank you for that. And so we will go into the next part of our presentation, which is Q&A, as you put in. And I would like to introduce James and also Sarah Diaz. Both of them will be moderating the questions from our audience here. And we have over probably 300, 350 people here, so.
All right, well, wonderful. First off, thank you guys both so much for coming. This was wonderful to hear the results. And again, I'm gonna back you up on that awesome animation in the chat too. Everybody was really excited to be able to see that in a visual form, because I think we all hear about it so often, but to really be able to appreciate it, what it does at the cell visually was really powerful. So I want to thank you first and foremost on that. To get us started, our first question kind of talks about the subgroups of ALS. So were there any subgroups on the trial participants who responded more favorably, either family onset, sporadic, fast progressors, or slow progressors? Did you notice anything?
Yes, so generally speaking, we enrolled fast progressors in the trial. That was by design. And this is because by doing that, we were able to have a relatively small and fast trial. Because we were able to see a treatment effect in that particular population that is predicted to progress at a fast rate. In terms of subgroups, we didn't see anything that is clear at this point. However, I do want to say that we are doing additional analysis, specifically looking at genetic status. So we collected DNA on an optional basis. And the analysis is still ongoing. So that's going to be quite interesting. And we did save extra samples also in terms of plasma samples. And so it's possible that perhaps we might do some additional analysis with respect to that specific biomarkers. But again, all of this is still ongoing.
I would say that all of that's really important, but it's still a relatively small study. And there's going to be, I think, limited kind of number of subgroups we can do and say something with a lot of confidence. But certainly to explore and lead to other ideas, it'd be definitely worth doing.
Hi, James here. Once again, amazing presentation. Going right into the questions because we got quite a bit. What were the eligibility parameters for the patients in the trial?
Yeah, so we designed the trial based on very detailed statistical modeling. And this was based on a database called PROACT, which is a great tool in the field. So by doing that, we were able to analyze data from 10,000 patients with ALS who participated in previous trials. And so we were able to identify some key features that would predict fast progression in those individuals. Specifically, the two main inclusion criteria that I want to emphasize are that for this trial, we enrolled people with definite ALS by ALS Correal criteria. These criteria are commonly used criteria to diagnose ALS. And when we say definite, what we mean is that the disease was already present in at least 3 out of 4 body areas. So we basically divide the body into 4 regions: the face, the upper body, the arms, the legs, and then the trunk and respiratory area. And so in order to be enrolled in the trial, people needed to have evidence of disease in at least 3 areas, meaning that the disease was already spread. and they had to be within 18 months from the first symptoms of weakness. So sort of relatively early in the disease.
Wonderful. Okay. In terms of phenylbutyrate, what was the idea or what is the mechanism behind it that led to the thought that it may work within ALS? This may be a question for the for Justin and Jenny. They came up with the drug.
Happy to say a little bit, and then maybe Justin or Josh can. It's something called HDAC inhibitor, and there's a series of these drugs that have been tried in models of neurodegeneration, including ALS and Huntington's and other models, and shown to protect neurons. And so that, again, they work by helping your body take care of kind of mismade or misfolded proteins. But there's actually been a lot of preclinical work suggesting that that whole class of drugs could be protective for motor neurons. And this is probably one of them that has good human data and good safety profile and gets into the brain. I know, Josh, if Justin, you wanted to add to that.
Yeah, certainly, and actually, we found out later that Dr. Cudkowicz had studied phenylbutyrate with her colleagues over a number of years as an individual agent and as a potential therapy for ALS. So the real rationale, going back to the mechanism, was we thought that simultaneously targeting mitochondrial dysfunction and endoplasmic reticulum stress pathways would be really important. And given that there are so many different things going on in ALS, we wanted to have a very broad approach. And so phenylbutyrate targets the endoplasmic reticulum stress pathways. The ER, as it's called, is the quality control center of the cell. So it helps proteins fold. And in ALS, we see all sorts of misfolded, mislocalized proteins. And so what phenylbutyrate does is to help with that process to make sure that the proteins fold properly and that the stress response in the cell goes back to normal. So that was the rationale for it as an individual drug. The reason we did the combination is so we could target two sets of distinct pathways at the same time, thereby we hoped having a stronger effect than either alone, which is what we saw in all of our preclinical studies, that both drugs were individually active, but the combination was much stronger than either drug alone.
Thank you, Justin.
To go back to the trial, were vent-dependent patients allowed in the trial? And do you believe there will be progress seen in vent-dependent patients as a result?
So if people developed ventilator dependence throughout the trial, they were kept in the trial. So at entry, people had to have a vital capacity of at least 60%. Again, based on statistical modeling. So, I would assume that, most people did not, obviously, if they had vital capacity of at least 60% or predicted, they were not vent dependent. However, we did have people who developed ventilator dependency throughout the course of the trial. And so as long as they wanted to continue and they were medically stable to continue, they were allowed to stay in the trial.
In terms of, we know you talked about how patients could be on other medications, but could patients also be on supplements during the trial? Other supplements over the counter?
Yeah, people could not be on participating in another active trial at the same time, which as is common for ALS trials, where you don't want people to participate in two trials at the same time, although they can participate sequentially. So general supplements for wellness were allowed. We had a very short list of medications that were not allowed in the trial.
Wonderful.
And what differences did you see with the percentage of responders in the initial trial versus the open label extension?
So I don't know that this was not specifically a responder analysis. Again, we looked at the effect on the entire population. So we just measure different things. In the randomized period, which was a short period, 6 months, we were able to look at differences in function, because essentially we would ask the same questionnaire multiple times every few weeks to everyone who was in the trial. And then long term, we measure survival in the entire population. Again, this was not just based on a few responders. It was an effect on the entire population.
Did any of the trial participants experience any sort of reversals or nerve reinnervations?
So the results I presented today are based on sort of as commonly the case in ALS trials. That's the average response. So obviously there was a range, and we do have some graphs that we didn't present where you can see sort of some variability with patients maybe experiencing more effects. However, I do want to point out that, as you know, ALS is a very variable disease, and no two patients are the same. So it's very hard to tell kind of, exactly, to really make those conclusions, because what's really important is the average response, because people can have some variability when you compare two individuals.
Just to stick on the trial as well, did the participants know the 2-to-1 ratio of AMX0035 to the placebo?
Yes, it was publicly available. The trial design and sort of inclusion criteria, et cetera, are published on clinicaltrials.gov. So that's something that, it's common practice in all our trials.
Also in the consent form, when people sign up, it tells you what the ratio of placebo to active is for every trial.
Well, I want to say thank you for doing the 2-to-1. That is such a huge progress for ALS patients in the trial setup. So, we are very grateful for that, that trial design. In terms of next steps and kind of the roadmap for getting this drug available, because we have a lot of patients on the call tonight that are kind of wondering, well, we saw so much great outcomes. When can we maybe see this as available to a larger population?
Yeah, maybe I could ask Justin and Josh to explain. I know we're all working together closely to take the next steps, and maybe Justin and Josh can elaborate on that.
Sure. Yeah. So that's what we work on every single day is trying to get this drug forward as quickly as possible. So what we've been doing is we've been working collaboratively with regulatory agencies worldwide. So FDA, but not just FDA, also in other countries and areas as well. And I think our goal as we've been going about it is just realizing that every day, every hour counts, and trying to make sure that comes into the conversation when we're talking, with the regulators and otherwise. But both U.S. and other countries do have a very rigorous, drug evaluation and approval process, and so we're trying to go through it as quickly as we possibly can. But it is a bit of a process, and we're hoping we can do that collaboratively, with the agencies. And in parallel, we're also talking to the agencies about different, possible expanded access programs. We don't have anything definitive yet, and it will ultimately, depend on those interactions. But it's something we're thinking a lot about, and really, what we're focused every day is just, shaving time off the clock and seeing if we can get this forward quicker.
Wonderful, thank you.
Always good news to hear. Can AMX0035 help ALS patients specifically with behavioral variant FTD? And if not, are there any other treatments to help or slow it down?
That's a great question. So we didn't specifically measure cognitive or FTD-like symptoms in the trial. I agree it would be interesting to look, if when the drug becomes more available, or in other studies, it would be good to look at the effects specifically. So ALS and the cognitive symptoms are part of a spectrum, and that's caused essentially by the loss of nerve cells in different parts of the brain. So if the mechanisms are the same, I guess one could hypothesize that the same drugs might help slow down the loss of different neuronal pools And so, it's not, again, it's possible that it might help, but we didn't formally test it as part of this trial.
So it's different biology. I think maybe everybody knows that Amylyx is also doing a study of the same combination in Alzheimer's disease. That's what they were initially planning until we kind of sidetracked them into ALS. they did go back to doing that as well. So I think there's going to be some use for this for the cognitive parts and for other neurodegenerative diseases, but those studies have to happen first.
Yeah, and mechanistically, and by the way, Merritt, that was the most wonderful sidetrack of our lives. We're really honored to be a part of the community here. And the goal of the therapy, as Sabrina said, is to protect the nervous system. And so we think that in any disease with ongoing neurodegeneration, that the drug may have benefit. We don't know that though until we run the proper studies. And so we have the ongoing Alzheimer's study, and we'll be following up with FTD and other neurodegenerative diseases as well.
Well, I know everyone on this call is happy that you guys got sidetracked over to ALS. So we'll take that as a win for our disease process. I know you discussed during your talk, Sabrina, about the changes that you saw with upper motor neurons. Would that be with the highest locations in the FRS scores or decrease in how fast they were dropping? Or did you also see it in bulbar symptoms and lower as well?
Yeah, so basically the ALS FRSR calculates a total score, which is essentially a total body score. But then you can really look as an exploratory analysis at different parts of the body. And the 4 areas are upper body, lower body, respiration, and bulbar. And by bulbar, I mean speech and swallowing and salivation. So essentially, you can look at each subdomain to try to see if there was a more pronounced effect in any of these body parts. Now, again, sort of similar to the discussion we just had, because the mechanisms of disease are sort of the same, and then you just had one area that can be affected first or more in individual patients. The thought is that all areas in theory could respond. Now, what we did see is that all the 4 subdomains moved in favor of AMX0035. And again, we had a statistically significant and clinically meaningful result when we looked at the total score, which is what's validated essentially and what the study was designed to detect. Now, as exploratory analysis, we also looked to see if there was any area of the body that moved the most. And the good news is that they all moved in the right direction. If you look numerically and you really want to look again at the numbers, the upper body moved a little bit more than the other parts of the body. But again, that's also known that the upper body subscore or subdomain is the one that contributes the most to change over time. It's just a function of the way the questionnaire was developed. So if you do longitudinal studies and you look at the entire population, when you look at progression on the score, on the total score, 40% of that progression is due to progression in the upper body scores, because they capture more fine activities where it's easier to see a change over time, as opposed to, let's say, for example, other parts of the body, like the respiration, for example, depends a lot on BiPAP use that can vary due to other reasons. So again, it's the most sensitive to change, and that's the one that moved the most.
We have a lot of eager people here tonight, so I'd be remiss if I didn't ask. Will there be a phase 3? And if so, will there be an open enrollment as well?
No, it's a great question, and we don't have an answer yet, which is the certainly frustrating. But as soon as we do, we'll let everyone know. The reason I say that, though, I think is a good thing. The paths before us are either a path towards approval, in which case, in the meantime, we'll do an expanded access compassionate use program, or there will be a phase 3 trial, in which case certainly our goal is to enroll as quickly as possible at as many centers as possible. The reason we don't know is because that's really a question for the FDA. And I shouldn't just say the FDA, I think for the EMA and Health Canada and regulatory bodies generally. And so we don't have those answers from them yet, but as soon as we do, we'll let everyone know. And I think our biggest goal is access. And so we think as soon as we can get those answers through either path, we can get access as quickly as possible.
Great to hear.
Awesome. In order to be able to respect your guys' time, we will be able to ask probably one more question. And this is a big question we've gotten a lot. Unfortunately, ALS care can be very expensive. And so what are the thoughts on the price of this and potentially for the future of insurance companies covering it, or kind of your thoughts on helping the community afford expensive drugs?
So we haven't made any decisions on pricing yet. So we're still learning about that and trying to figure out as best we can. I think our kind of guiding principle as we've been thinking about it is we never want price to impede access for any individual patient, and that's going to be kind of our true north as we think about it. But on the other side, I think we do want to make sure our company can continue moving forward and growing, and hopefully making further and greater innovations in ALS and otherwise. So I think it's a bit of a balance, but I think the first aspect that we think about is we never want price to impede access to even a single patient.
And I think too, to just add on to that a little bit, to Merritt's earlier point, I think that we feel really just so honored to be part of the community and to get to talk with all of you and work with people like Dr. Paganoni and Dr. Cudkowicz, and so and into you as well. And so a huge part of our goal is to build our company around how can we support the ALS community, how can we be a partner. And so the company's not just about, hey, we have a potential therapy, but how can we do much more? Because I think we feel very passionately that ALS has been unaddressed on many fronts for far too long. And if we're given the platform to do so, that's what we want to do.
I don't think that could have been better said. So thank you, Josh and Justin, for that. That was exactly what we needed to hear. I think on that, we will end our time. I want to thank so much for the 4 of you for coming on tonight and answering all of our questions and talking about your results. We look forward to future and where this is going, obviously in ALS, but also in other neurodegenerative disease processes. I think that's really important that we're looking at everything as a whole. You are more than welcome to stay after this part. We always have an open forum session to discuss kind of what everyone's mind, but we want to also respect your time. So thank you so much for joining us on this Monday night, and we will stop the recording.
Thank you very much, everybody.
Thank you all so much.
Thank you.
Thank you.
Thank you.